Welcome to this presentation on auxin regulation in Arabidopsis roots. This slide introduces the topic and frames our exploration of how auxin gradients and transport shape root form and function, setting the stage for a detailed look at molecular players, transport systems, and engineering opportunities to direct root architecture for research and agricultural goals. This slide explains how auxin is produced in shoots and root tips, then distributed through vascular tissues to create dynamic source sink patterns that guide development. The diagram highlights local production and long-range transport converging to form an auxin gradient across meristem, elongation, and maturation zones, and emphasizes how that gradient continuously reshapes root growth and cellular behavior. Here we see how pin proteins steer auxin streams by asymmetric localization, creating focused auxin maxima that define developmental outputs. The illustration links specific pin placements to maintenance of the stem cell niche and to gravitropic bending, showing that pin relocation and directional flow underlie root patterning and the ability to redirect growth in response to gravity. This page contrasts coordinated auxin influx in wild-type roots with impaired uptake in AUX1-LAX, loss of function mutants. In the normal root, influx through columella and protofloem sustains a robust auxin gradient supporting gravitropism and lateral roots, whereas impaired influx disrupts that gradient, reducing gravitropic response and lateral root formation with clear developmental consequences. ABCB transporters act as conditional auxin exporters and importers, modulating intracellular auxin levels and long-distance distribution. The central panel shows how TWD1 directs proper targeting of ABCB proteins, while the phenotype panel notes that double mutants display dwarf roots and impaired polar transport, revealing overlapping ABCB roles in maintaining hormone flows essential for root form. This schematic walks through the auxin signaling pathway from hormone perception to growth. Elevated auxin promotes TR1, AFB-mediated degradation of OX, IAA repressors, freeing ARF transcription factors to activate target genes, which then drive cell cycle progression, cell expansion, and root hair differentiation, effectively translating hormone dynamics into concrete cellular and developmental responses. Auxin response factors, or ARFs, tailor gene activation to environmental and developmental cues in roots. The left diagram links specific ARFs to root hair elongation and lateral rooting under particular conditions, while the right panel shows how ARF5-MP stabilization in high auxin zones sharpens expression domains, giving precise spatial control over tissue patterning and organ-level outputs. This slide shows how moderate cold cues rewire auxin biosynthesis and transport to elongate root hairs adaptively. Cold elevates TAA1, UCK activity, and local IAA production, which is then redistributed by PIN2 and AUX1 to promote root hair growth, enhancing nutrient foraging under suboptimal temperatures, and illustrating a hormone-mediated adaptation to environmental stress. The diagram describes bidirectional coordination between shoot-perceived light and root auxin distribution. Light signals in the shoot modify transcriptional networks that boost PIN and AUX1 expression, promoting lateral root branching, while auxin exported from the shoot can suppress root greening via downregulation of greening regulators, demonstrating coordinated signaling across organs to balance growth and resource allocation. This slide outlines a precision engineering pipeline for auxin-regulated root architectures using genetic tools and synthetic circuits. By combining CRISPR promoters, auxin-responsive devices, PN and ARF control, and real-time biosensing, we can achieve spatiotemporal control of hormone gradients, produce predictable root systems optimized for stressors, and guide breeding strategies to improve phosphate uptake and drought avoidance. Translational potential is highlighted by two strategies: enhancing auxin export for example via ABCB19 overexpression, can deepen rooting depth and improve drought tolerance, while attenuating ARF7 signaling can steer steeper root angles suited to dense soils and efficient nutrient foraging. Together, these mechanistic insights point toward climate-smart crop improvements and resource use optimization. The final slide presents an integrated Auxin regulatory framework linking core mechanisms, polar transport, receptor sensing, and ARF outputs, with environmental network rewiring and a crop engineering blueprint. This systems view identifies tunable regulatory nodes for biotechnology and outlines how understanding and rewiring these layers can produce resilient, resource-efficient crops for future agricultural challenges.